Good morning. Thank you for staying to the last minute. I'll be talking about mathematical models of RNA expression profiles, potential application to drug discovery and personalized medicine. The key idea behind my talk traces back to these two people, Paracelsus, 1493 to 1541, and Prigogine, 1917 to 1541. Paracelsus said that the dose makes the poison, which is familiar to us. Paracelsus said that the dose makes the poison, which is really related theoretically to the statement made by Prigogine, who says life is dissipative structures. So behind the dose makes the poison is the concept of dissipative structures. We know that drugs, unless it reaches a certain dose, will have no effect. Any drug which goes beyond a certain level will have toxic consequences. The maintaining blood level of drug is a dissipative structure because the drug level is determined by the input and output, and both input and output processes involves active process, active transport, which requires energy. So anything that exists because of the dissipation of free energy is defined by dissipative structures. So anything that can exist without dissipating energy is called equilibrium structures. So Prigogine defines everything in the universe into two groups, equilibrium structures and dissipative structures. And I think there are three main types of approach to drug development. One we may call top-down approach, for example, herbal medicine, practiced by millennia by humanity, and the discovery rate of that may be 1 out of a million. This is my guess. The other one is the bottom-up approach. Molecular pharmacology is a good example of that. And we know that the success rate of this approach is 1 out of 100,000. The new approach that I'm advocating, beginning this meeting actually, is maybe called the hybrid approach or complementary approach, which combines both top-down and bottom-up approach. So these three approaches can be characterized in terms of two theoretical concepts, equilibrium structures and dissipative structures. Top-down approach emphasizes dissipative structures, okay, life itself. And bottom-up approach emphasize the equilibrium structures, studying the 3D structures of the receptors, 3D structures of the proteins, enzymes, and so on, which can exist without dissipating energy. So that is the equilibrium structures. Of course, even bottom-up approach heavily depends on the dissipative structures at the clinical trial level. So this is maybe an exaggeration, but I'm trying to emphasize the fact that these two theoretical concepts may be useful in distinguishing these three approaches, and the hybrid approach involves both, emphasize using both equilibrium structures and dissipative structures, particularly this one, which is not widely recognized. So we have to know something more about dissipative structures. What are dissipative structures? Dissipative structures are in physics, chemistry, and biology. In physics, the electronic transitions occurring in atoms to give you light, coupled to chemical reactions, is the dissipative structures, because these dissipative structures will disappear when chemical reaction stops. No energy supply is there. In chemistry, the well-known Belousov-Zhabotinsky reaction, this is a picture taken of Petri dishes from the top. Each Petri dish contains maybe a dozen different chemicals, some organic, some inorganic, and the reaction products are visible, in this case carbon dioxide, and each frame is taken after about a minute, going this way, and you can see the pattern of bubbles forming and merging and interacting. And these are structures. These are carbon dioxide concentration gradients, which will stop when the reaction stops. So this is a dissipative structure in chemistry. And dissipative structure in cell biology was first, I think, measured in 1984 by a couple of scientists at the University of Virginia. And these are neutrophil migrating toward an opsonized particle. And if we concentrate on this column, the colors are proportional to the intracellular calcium concentration. In the beginning, calcium ions are low, and as it moves, the calcium concentration is high in the moving forward part of the cell and low at the tail. And these concentration gradients is what causes this cell to move. And concentration gradient will disappear if the cell runs out of energy. So this is a good example of this structure inside the cell. And in 1983, I was invited to give a talk in an international conference organized by R.K. Mishra at the All India Institute of Medical Sciences in New Delhi. And while preparing for my talk there, I came up with a model of the cell because the concept of dissipative structure helped me organize everything from DNA to RNA to enzymes to chemical reactions, and all these are necessary to form right concentration gradients inside the cell. It could be calcium ion, it could be proteins or potassium and so on. These dissipative structures of precogenes are the immediate cause of cell function. So this model was called Bopolator as a result. And so dissipative structures are at the center of the theoretical model of the cell, which is the basic building blocks of all living systems. So all the drugs that we develop has to interact and affect the function of the cell. That means it has to affect dissipative structures. Otherwise, you will not have any effect as a drug. So the overall strategy for applying this theoretical concept to developing drug involves two more things, not just the theoretical concept of dissipative structures, but experimental tool provided by the microarray technology invented at Stanford in 1995. And then there is a mathematical tool that I'll discuss, which are, there are a couple. And all these can be integrated in a scheme like this. So I'll give you the overall view, and we'll deal with some of these details as far as the time allows. So microarray technique is the key to drug development, in my opinion. So microarray technique, as you know, allows us to measure thousands of RNA concentrations in living cells simultaneously. So we can measure RNA levels in cells, whether control cells or tumor cells before giving anti-cancer drug, or tumor cells after giving anti-tumor agents. So we've got 3 samples. We can generate RNA data from 3 samples. In this case, I have data from 20 breast cancer patients measured by Peru et al. At Stanford in year 2000. And from these 3 sets of data, we can generate 2 numbers: RNA changes due to tumor, RNA changes due to drug. These 2 numbers can be converted into 1 number by taking the ratio of the 2 and calculating arctangent. So these two numbers can be converted into angle. And this angle can be converted into 9 different mechanisms using mechanism circle, as I'll show you. And these 9 mechanisms generates mechanisms table. And this table allows us to identify beneficial and harmful gene expression profiles. Not genes themselves, but profiles. Okay? Each harmful, beneficial, and harmful expression profiles can be formed by different sets of genes, not fixed set of genes. And these two expression profiles allows us to calculate group micro-therapeutic index for the drug and population or personalized micro-therapeutic index of the drug. And this will allow us to do personalized medicine, and this will allow us to do a discovery. So if you have patient X, you can do biopsy to have a normal tissue or tissue culture, and tumor sample, and tumor sample after drug treatment, or this tumor sample can be cultured in vitro in the presence of drug to generate AF prime sample. For this talk, I'll only dwell on these 3. So RNA measurements lead to this kind of graph. This is the tumor RNA level in tumor tissue minus RNA level in control. And if there's no difference, you have 0 value. 1-fold increase will be this, 2-fold increase, 3-fold increase, or 1-fold decrease, and so on. And these x-axis represent patient number, 20 patients. And each line is 1 RNA encoded by 1 gene. And we have data for about 5,000 genes, so we'll have 5,000 traces like that altogether. And this is normal tumor tissue minus control before giving drug, and this is signals from tumor tissues after giving drug minus tumor tissue before drug. And from this original data, we can identify some mechanisms. If the RNA level increases due to tumor, it will be plotted along this axis. If it decreases due to tumor, it will be on this axis. If drug causes increase in RNA level, it will go up, and decrease will go down. And the 9 is those profiles which is not affected either by tumor or drug. So we can calculate the angle alpha for each patient, for each patient, and if alpha is plus minus, say, 10-20% of 0, we call that mechanism 1. If alpha is greater than, say, 10 degrees and less than 80 degrees, we call that mechanism 2, etc. So you can go around the mechanism circle like that. And this is a tabular form of what I just said. So angle value and the mechanism. Increase due to tumor, no change by drug, that is mechanism 1, and increase due to tumor, and increase due to drug, that's mechanism 2, etc. And for example, this mechanism 2 is obviously harmful because RNA level is enhanced by drug as well as the tumor. So tumor and drug has same directional change, so we call mechanism 2 as harmful. Whereas mechanism 4, for example, involves RNA levels in tumor going down. In other words, RNA levels decreased by tumor, but increased by drug. So that we could speculate to be beneficial to the patient. So we can divide these mechanisms to beneficial and harmful mechanisms. This table summarizes all the information we can get from the RNA measurement from different patients. So for a given patient, say 1, 2, 3, for patient 1, we have data from normal tissue, normal sample, before sample, after sample. From that, we determine mechanism. And we can do this for each of the 9,000 genes, or in our case, only 5,000 of them. And from this, we can calculate individualized or personalized micro-therapeutic index, and survival month is measured, and this is the filtered micro-therapeutic index, which I will explain shortly. So all the information from the RNA measurement from various patients are included here. And this is another way to represent some of the information in that table. Again, we have a gene here, 4,740 of them, and patient number. And each gene exhibits beneficial profile or harmful profile in different patients. In this patient, this gene expresses beneficial profile. Here, harmful profile, and so on. And we can count the number of beneficial profile and harmful profile exhibited by a given gene in different patients. So sum of these two will be 20. This table has a lot of information, a lot of important message for us. For example, for a given patient, there are genes which exhibit beneficial behavior and harmful behavior. That's not surprising. But what surprised me is actually a given gene can be beneficial or harmful to different patients. Gene expression profile. This is because before we started this experiment, we had a hypothesis. We thought that by measuring the RNA levels from these breast cancer patients, we should be able to identify a set of genes which is associated with breast cancer, okay, regardless of who the individuals are. But my students and I were very surprised, and I wasn't surprised, but disappointed, actually. More my student than myself. I got out of that disappointment right away because I saw the theoretical possibilities here. So the message that I got out of this data is this. The traditional view is that there's a common set of genes responsible for tumor. Breast tumor, kidney tumor, pancreatic tumor, and so on. That is the predominant view of oncogenesis community. But what if it's not the case? What if it's the phenotype or RNA profile, expression profile, which is a phenotype, molecular phenotype, right? What if it's the molecular phenotypes which are responsible for cancer, regardless of what genes generate such a phenotype? And if that is the case, we are going from equilibrium structures to disparate structures. DNA sequences, gene sequences are equilibrium structures. And the RNA profiles, gene expression profile or RNA profiles, they are equilibrium structures. So instead of looking for the cause of cancer at the gene equilibrium structures, what if we look for it in the dissipative structures? See? This is where the Paracelsus and Prigoghine's idea comes in. According to them, life is dissipative structures. According to Paracelsus, toxins are dissipative structures. What if drugs are also, well, drug effects are also dissipative structures? So in this table, you can calculate personalized micro-therapeutic index, which is simply, for this person, you'd simply count the green boxes and divide that by number of red boxes. That will give us the micro-therapeutic index personalized for that patient. So in this patient, the drug doxorubicin has 4.34 micro-therapeutic index. And in this patient, you do the same calculation, you get 2.94, and so on. So each person has their own micro-therapeutic index. Yes?
This particular chart that we have is specific for the drug doxorubicin.
Yes, yes.
That means for every drug that I am going to give to my patient, I need to develop a similar chart.
That's an excellent point, yes. That's what I think.
But given the fact that cancer therapy is a multi-drug therapy,
Uh-huh.
and we see that they are done with loads of drugs,
Yeah.
at toxic levels and various levels, so don't you think the whole structure of this chart will get more and more complex?
Exactly, yes. This is just for doxorubicin. Yeah. We can generate same table for all the anti-tumor agents or breast cancer drugs.
Yeah, but at the same time, it also takes into consideration the patient.
Yes.
That means,
Personalized medicine. We have to do personalized medicine. Yes.
which is what we don't have at all.
No. Yeah, this is what I call hybrid approach to drug discovery. Yes. And on this side, you can count the number of green boxes for the whole set of genes to calculate the population microtherapeutic index. Okay? So this population therapeutic index would be useful in characterizing the drug. And microtherapeutic index would be essential for identifying the right drug for right patient. That's what I mean, personalized medicine and drug discovery research. Poisson distribution is useful for summarizing experimental data. Poisson distribution has one parameter, lambda, and it is a function of number of events, k. If you know how many events occur within certain time window, say 10, this equation will allow you to predict the probability of that event occurring, say 5 times within that same time window, or 15 times. It's a symmetric. This equation, it was found to fit our data. So beneficial and harmful mechanisms fit the Poisson distribution. This is the number of patients on the x-axis in which the RNA expression profiles are beneficial in 10 patients, in 5 patients, 7 patients, and so on. So experimental data, which fits Poisson equation, theoretical equation, with lambda value, in this case, of 5, around 5, meaning that on average, this doxorubicin has a beneficial RNA expression profile in 5 patients. And similarly, the lambda value for the harmful mechanism is about 2. So this means that the doxorubicin has harmful RNA expression profile in 2 patients on average. And this ratio would be microtherapeutic index. So we had a hypothesis. So those patients having high microtherapeutic index should survive longer than those patients having low micro-therapeutic index, okay, if our theory is right. So we plotted survival month here and micro-therapeutic index on the axis, and we got all this scatter. Again, we were disappointed, right? We anticipated something like this, but we got this instead. And again, my students were more disappointed than I, actually. He almost gave up. He's a PharmD student, Larry Cheng, and there are two students, actually, Larry Cheng and David Yao at Rutgers School of Pharmacy. But I think to expect a linear correlation between micro-therapeutic index and survival month may be too naive. Because different patients may die because of different mechanisms, not because of the RNA expression profiles, okay? So it may be possible that we can divide patients into three groups: those who survive much longer than predicted, and those people who survives as predicted, and those people who live shorter than predicted. Three groups. So if we can come up with some way of identifying these three groups of patients, maybe we can have this straight line and get some kind of a FDA approval for this approach. And one way to look into the characteristics of individual patients, the RNA profiles of individual patients, is provided by an equation that traces its origin to blackbody radiation equation, which I don't have time to go into, but you can look it up on my homepage website. If you go to my homepage and click on publications and book chapters and chapter 11 and 12, I have explained it in great detail. So to make the long story short, blackbody radiation equation generates a spectra, blackbody radiation spectra, and Max Planck discovered this equation that fits the spectra. Now, when I saw this figure, this blue line, in a paper published in 1998, I think it was, by Liu et al., which is a histogram of single molecule enzyme turnover time, okay? That curve looked very similar to blackbody radiation spectra, so I used this equation, tried to fit the data. And that is shown in the red line. Because of the noise, it's sort of difficult to see whether it really fits or not, but my confidence in the applicability of the equation that fit this equation came from its ability to fit so many different biological data. As long as it has a fat tail, it fits. So far, I've tried about 25 of them, okay? I don't have time to show you here. And the reason the enzyme kinetic data and blackbody radiation spectra fit the same equation, same type of equation, I think it's because of the quantization of energy levels in both systems. In atoms, energies are quantized into atomic orbitals. In cells or enzymes, the energy levels may be also quantized. As long as you have discrete levels of energy, this equation will fit. And this equation that I'm talking about looks like this. In the dependent variable y is a function of x, and it has 4 parameters: small a, small b, capital B. And this reduces to Planck's equation when a is some products of a universal constant, capital A is 1, B is 0. Okay? Then it reduces to Planck equation. That means this equation fits blackbody radiation spectra itself. It fits almost exactly. And the reason this equation fits so many data, I think, is because of the wave-particle duality that we all learn from high school physics. Okay, people think particle-wave duality only applies to physics, but since this equation is the source of that concept, and this equation has two parts. First part indicates the number of standing waves per unit frequency per unit volume, no matter what the system is, if it's a single enzyme or whole cells or whole brain. It's a volume. Within that volume, there may be standing waves, okay? Depending on the energy level of the system, you have different number of standing waves. And this portion tells us the average energy of these standing waves, okay? And this is a power law here. Variable is raised to a certain power, and this is an exponential function. Variable appears as an exponent. So these two products has a very powerful effect. It accommodates all fat-tailed distribution, okay? And this is a picture. Standing waves. 1, 2, 3, 4, 5 standing waves are in this volume. And depending on the amount of energy you put, you can increase the number of standing waves. And you can calculate the average energy of these standing waves using quantum mechanics. And these waves, the number of waves, determines blackbody radiation spectra, protein folding free energy, enzyme catalysis, turnover time, RNA levels, which I'll show you shortly, and the RNA levels in cancer patients and RNA levels in single cells, and T cell receptor variable region gene diversity, functional magnetic resonance imaging signals, and the decision time histogram of all things, brain functions, and word length distribution in text. And polarization of the microwave background radiation that people claim to have measured in July also fits this equation. All these implicates, I think, wave phenomena. Okay. This is, again, that equation, which I call Planck distribution. This was discovered in 2008 at Rutgers University. And this is RNA levels in normal human breast tissue, for example. Experimental and Planckian distribution, this is Gaussian. So anytime you have this fat tail, it seems to fit Planckian equation. So it goes with the Gaussian, okay? So we apply this equation to several well-known metabolic pathways or protein families in human breast tissues. And this is some of the results, experimental and theoretical. BRA stands for blackbody radiation-like equation, which was the previous name. I decided to make it shorter, Planck distribution. You see, the fit is very good. Yeah. The one application of the fitting of the cancer data to Planck distribution is to characterize each metabolic pathway or each RNA expression profile in terms of 3 parameter values. B over A, capital B over B, small b, capital B over capital A. But we found that small b over A is most useful. It varies from different metabolic pathways to different metabolic pathways, and these others are not as sensitive to that. So if we concentrate on B over A value of this RNA profile, family of RNAs, before and after giving drug, the B over A value changes from 3.539 to 3.42, and the p-value is 0.14. So this has no effect. So this ratio doesn't change between before and after samples. But here, in this metabolic pathway, B over A value changed from 2.6 to 2.7868, and the p-value is 0.02. So this difference is significant, and so is this. So this is borderline. This is different. So this ratio can detect the effect of drug treatment on certain metabolic pathway, but not all pathway. Okay. And this table shows the B over A ratio values of different pathways. And as you can see, this pathway is different from that pathway. P-value is 1.5 times 10 to the minus 3. So all of these values, which is symmetric with this, are less than 0.05, indicating that B over A ratio can distinguish among these different metabolic pathways. And this may be useful for cancer research. So using this method, we may be able to identify those patients belonging to the upper or middle or lower levels in the survival month graph that I showed you. So to conclude, the microarray technique or its equivalent, when used in combination with mathematical tools such as Poisson and Planckian distributions, will enable biomedical scientists to discover anti-cancer drugs without knowing detailed underlying molecular mechanisms. The same microarray-based method can be utilized to identify the most efficacious anti-cancer drugs for individual patients. And there are no genes uniquely responsible for the tumorigenesis or metastasis. Hence, no single anti-cancer drug exists that can be applied to all cancer patients. Hence, personalized medicine is inevitable. Thank you very much.